All right, continuing with Module 6, we're going to now talk about influenza, which I'm sure many of you are familiar with. Some of you have probably had. Some of you have probably had the vaccination, so it shouldn't be completely foreign to you. But there have been some serious epidemics, pandemics, whatever, in the past, and we may be facing another one soon. So good to know about. So the causative agent is pretty simple. It's the influenza virus, which was first isolated in 1940. It's single-stranded RNA. It's got a capsule. There are 4 genera, which are called types, A, B, C, and D. And pretty much A and B are clinically relevant for humans. The virus has surface glycoproteins, and this is important, so remember this. They have hemagglutinin, which is abbreviated H or HA, and neuraminidase. We abbreviated N or NA. And the influenza virus is very mutatable. They alter their nucleic acid pretty often, which then alters their outer proteins, the hemagglutinin and the neuraminidase. And we'll see some reasons why that's important as we go on. So here's the influenza virus structure. Here's the nucleoprotein, which is RNA. It's got an envelope, which is right here. It's got the capsid, which is over here. And then here are the surface proteins, neuraminidase and hemagglutinin. I think they're representing purple here by hemagglutinin and the white by neuraminidase. That's less important, but just do know those. So influenza A viruses can also infect a wide number of animals. So you can see the full list here, but the most important ones to remember are pigs and birds of all kinds. And birds are a primary reservoir for influenza A viruses. And viral influenza B pretty much just affects man and pigs to a certain extent, and they've just discovered it can infect seals, but those aren't going to be very important as we talk about influenza. It's the influenza A that's the serious one. So influenza A, its most outstanding characteristic is its rapid evolution. So that means it's going to have lots of different forms that are circulating in the world at any given time. It's divided into subtypes. As I mentioned, there's 16 different H or HA subtypes and 9 different NA or N subtypes. And that can change at any time, and we can add more. But currently, right now, there are no influenza B subtypes. It's just influenza B. So the reason for this variety in influenza are 2 things. One is called antigenic drift, and one is called antigenic shift. But antigenic drift, RNA virus, as I mentioned, have a high mutation rate. DNA viruses don't. So the mutations which occur cause a slight variation or a small mutation in the surface protein. That's called antigenic drift. And you can see here we have some little purple surface proteins, and then over here we have some purple surface proteins, but they've changed somewhat. They're still purple, and they're still somewhat of an oval or partly oval shape, but they are slightly different. So that means with drift, small changes, that means the viruses are closely related. But they do change frequently. The body's immune system may or may not recognize them. If they've changed just a little bit, you probably will. It depends on how much they've changed as to how different they are, which means your immune system may or may not recognize them. And antigenic drift is the reason why people can get the flu more than once. The common knowledge among microbiologists is that you can pretty much only get a virus once. But if a virus changes or mutates, that means you can get it again. So we saw that with the coronavirus. We have some antigenic drift in the coronavirus as well. So flu vaccines they review the circulating strains of flu each year, so that would be different H and different N protein spikes, and then they create the vaccine based on what's currently circulating. Excuse me. So here's just another image of antigenic drift. I've got a few pictures of this because sometimes it's hard to grasp the first time. So here's virus A, and then there's some mutations over time, and then we've got a change. Here's a little change in the nucleotide, the RNA, which then causes the surface proteins to change slightly. Here we have orange and purple. We have circle, orange circle, purple diamond, and here we have orange circle but purple square. So it's changed a little bit. So if your body has already made antibodies to this orange protein spike, then you likely will be protected. So an antigenic shift is an abrupt major change. So there are two ways that a shift can occur, and that's when a flu virus comes from an animal and it gets the ability somehow, and that's by, evolution or, mutation, to infect humans. And so these animal origin viruses have completely different H or HN combinations. So that means they're a novel virus, meaning a brand new virus. We haven't seen it before. So that's the first way. We can have an antigenic shift in a virus coming from an animal. The second way is if two different viruses simultaneously infect the same cell. And this is typically in an intermediate host. For example, a pig might get a human influenza virus, and it might get an avian influenza virus. They're in there together, they're reproducing, and they might resort their nucleic acid. They might get it mixed up and get RNA segments from each parent. And that's going to create a completely new influenza virus. So that's going to be a new H, a new A, or a new HN combination. That's again called antigenic shift. And you can see here, this one actually has 3 different types of influenza A in it. And we'll say this is the pig. They've got some green dots, purple dots, and yellow dots. And then the new strain has all 3 dots. And we've never seen that before, so that's going to cause some problems. And that's going to create a flu pandemic. And a flu pandemic typically occurs every 10 to 40 years. Depends on, when this gets into an animal, when they resort, how strong of a mutation it is, etc. So here's another graphic about antigenic shift. We have virus A, we have virus A here, 2 different viral A's. They're in a single host. They mix up the RNA. You can see that here. Here's some blue, here's some red, here's some red, here's some blue. And then it has surface proteins that look like neither of the parent. This one has blue circles, this one has blue squares. This one has purple diamonds or triangles, and this one has purple circles, purple globes. So entirely new virus C. And here are the 2, again, just so you can get a full grasp on it. Here we have antigenic drift. So we have a yellow here, a full triangle, and a line. And here's a half triangle with a line. So that's drift. The same basic components, they're just slightly altered. And here with shift, we have something completely new. Okay, so now we'll talk a little bit about the history of influenza. It's been around a long, long time, and there are recorded descriptions of it throughout, modern history. We don't have anything on it in the old world, but, that doesn't really matter. It's probably been around then too. But a couple of earlier or later pandemics I'll mention just briefly. In 1729, there was a pandemic that started in Russia, and it spread through the entire world in 3 years, had very high death rates. There were several waves of infection. So the first wave didn't kill as many people. It had a lower mortality rate. And then people recovered, and then it spread around the world again, and then a new wave, just like we had with Corona. And the reason for that is because we're having antigenic drift. So it's mutating slightly as it goes around the world, and then it might become more virulent, more toxic, cause a higher mortality, which is what it did in Russia in the world in 1729. Then in 1781, we had one that started in China, then it went to Russia, and then to Europe, all in 8 months. The infection rate was really high, particularly young adults. And here's one stat, 30,000 fell ill each day in St. Petersburg. 2/3 of Rome got ill. It raged through Britain in the summer of 1782. So what are flu symptoms? We've got fever, chills, tiredness, muscle aches, runny nose, headache, watery eyes, sometimes vomiting and diarrhea. That's typically in children, and a dry cough. If you have a weakened immune system, you are susceptible to pneumonia. And this is why the young and the old or the elderly are more commonly getting more and more sick. It's more deadly to those groups, and that's because their immune system isn't quite as strong as yours. If you're in your 15s to 50s or 60s, you're pretty protected. All right, so when you get a bad case of flu, your immune system gets weakened. It's been fighting the virus and you've been quite ill. And then you get a bacterial infection, which is called a secondary infection. So here's a normal, oops, let me get my pen on. Here's a normal alveoli. It's all clear. There's lots of air space in there for gas transfer. When you get pneumonia, you've got a bunch of fluid and pus and bacteria in here, and it's not a good situation for airflow. So your lungs fill with fluid, and if you can't get rid of that bacteria, then you're basically just going to kind of drown. So again, here is a couple other pandemics. There was one in 1830 in China, spread south, and then another one, and then in North America, it hit North America in 1832. It recurred in Europe, and it had a high infection rate, but the mortality was not exceptionally high. So I've just talked about those few historical ones, but now we're going to talk about the really big one, and that one was really bad, and that hit the world in 1918. So some comments about it from people and physicians and journals. The greatest medical holocaust in history. The pandemic ranks with the plague of Justinian and the Black Death as one of the three most destructive human epidemics. Deaths in the hospital exceeded 25% per night during the peak. Not a good thing. So how did it start? It began in China, and it began with a genetic shift of the influenza. And the shifts are what typically trigger a pandemic because it's changed so much. So it created a novel virus that no one had seen before, and there was a loss of herd immunity. If no one has ever seen it before, then no one can protect the group. First appeared in the United States in Kansas at a military training camp, and then it spread throughout the U.S. from there. It spread amongst the soldiers who were in training camps and also who were traveling overseas. And by being in tight quarters on ships, they spread it even more. But few really paid much attention to it at that point in early 1918. You know, it was just like, okay, there's a few people who are getting kind of sick, but the first wave wasn't as serious also in this pandemic. But they were a sign of what was going to come in the fall and winter of 1918, which is when the flus typically are at their peak. So in the Great War, which was the war of the First World War, it erupted in the final stages of the World War. It started in several places kind of at once, but it was transmitted among men in the armies and aboard ships. So it was transmitted to Bordeaux, France in April, and then in May it went to the British Expeditionary Force, so they were fighting in the war, and then it reached Italy and Spain, and in Spain it killed 8 million people in 1 month, May of 1918. So that earned it its name of the Spanish flu. So the next waves, there were actually 4, but the first 3 in August 1918, it was traveling on crew members who were going from England to Sierra Leone. There were a few cycles of the disease in Sierra Leone, and then it mutated while it was there and caused a 10-fold increase in the death rate. The same time it hit the main port of France, there was another mutation that was markedly more virulent, and it spread throughout Europe. And then a ship arriving at Boston from Europe infected that entire city. And all 3 of those were much more lethal forms of influenza. And then other countries experienced 2nd, 3rd, and 4th waves that were all more deadly than the first wave. So again, here's the antigenic drift, small mutations. This was an antigenic shift. So we might have, people at that time might have been exposed to this, might have had immunity to this one, immunity to this one. Maybe not immunity to this one, but regardless, this one was so different that people could not fight it off. There was no immunity. Some physician comments: patients would rapidly develop the most vicious type of pneumonia that's ever been seen. Later, when cyanosis appeared in the patients, it is simply a struggle for air until they suffocate. So cyanosis is turning blue. So when your lips start turning blue and your fingers start turning blue, you're not getting enough oxygen. So then the third one: influenza patients died struggling to clear their airways of a blood-tinged froth that sometimes gushed from their nose and mouth. What a horrendous disease. One anecdote shared from 1918 was of 4 women who played bridge together late into one night, and the next morning 3 of them were dead from influenza. People on their way to work suddenly developed the flu and, like immediately went home or died at their workplace. Very, very quick deaths, somewhat like the Black Plague, but not the same, in many respects. Because there was a war going on, there was a severe lack of healthcare personnel. So most of the physicians were with the troops, and then they recruited medical students who were in their 3rd and 4th year to care for the sick influenza patients. And then those physicians who were still in the States, many of them died because they got influenza as well. The Red Cross recruited a lot more volunteers at home to help fight the influenza epidemic, and the nursing shortage was so acute, they asked businesses to allow workers to have a day off so that they could work in the hospitals at night to give the nurses some relief. And at this time, there were a lot more emergency hospitals that were created. Just a couple of notices about recruiting. This one: cooks, kitchen maids, clerks, housemaids, laundress, mortar drivers, school teachers, any others with knowledge of nursing, please help us. It was deadly. It was desperate. So public health measures were put in place similar to coronavirus. Stores couldn't sell. Funerals were limited to 15 minutes. Some towns required a signed certificate, meaning that they were virus-free, and railroads would not accept passengers without a certificate from their doctor in many cases, not all. There was ordinances that were put into place, like wearing masks and, quarantining at home and all of that. And if you did not follow those protocols, then you paid steep fines. And didn't do that during Corona, so hey. Bodies piled up because there were massive deaths, just like Corona and just like in the plague, although they weren't necessarily in, this was 1918, so they weren't typically buried, in the streets or anything like that. They made their way to morgues. But there was a shortage of coffins, morticians, and gravediggers because the demand was so great. And the public health department started distributing gauze masks as a means to help protect the population. Here's the Treasury Department, United States Public Health Service. Influenza is spread by droplets. Cover your cough and sneeze. Avoid crowds. If possible, walk to work. I don't know why that is. I guess because maybe they were taking more buses and trains than just walking or driving their own cars. Cars were very prevalent then. And avoid excess fatigue. If ill, go to bed. Here's some more information very similar to coronavirus. Anti-mask meeting tonight to protest against the unhealthy mask ordinance. So we know that that's kind of silly, but people then believed what they wanted to believe, and they do now as well. But you're getting educated, so you're going to help others to understand why it's important. And then here's a warning to mask slackers. And there were people, police or public employees walking around telling people to wear their masks, and mostly they complied. And here's some of the Red Cross workers making and distributing masks to be dispersed to the population in general. So let's look at some stats. Our usual influenza death rate, that's kind of any given year, is about 0.001%. It's usually elderly and young children. I think there's probably 20 to 50,000 people that die each year. And again, usually they're elderly and sometimes they're young children. The coronavirus we just experienced had a death rate of 0.3. So that's significantly higher than the typical influenza death rate. And that comes out to about 300 per 100,000, or 3 million versus the total population of 8 billion. Okay. In the 1918 influenza, the death rate was 3 to 5%. So that's 10 times as high as the coronavirus death rate. And the numbers, the actual numbers, were 2,230 per 100,000, or 40 million people died out of 2 billion. So 3 million people died with corona out of 8 billion, 40 million died out of 2 billion. So some really strong numbers there. It also hit the 15 to 30-year-olds really hard, 20 times higher than in previous years. And it actually influenza killed more than the troops of all countries that died in World War I. There were more deaths in 1 year of Spanish flu than in 4 of the Black Death, which occurred, as you recall, from 1347 to 1351. More deaths in 1 year than in all 4 of the Black Death. That's pretty remarkable. The effect was so severe that the average lifespan in the United States decreased by 10 years. So if the lifespan at that time, I think it was probably about 45 or 50, it went down to 35 to 40. It was cited as the most devastating epidemic in recorded world history. 1/4 of the United States and 1/5 of the entire world was infected. President Woodrow Wilson, he got the flu, and he was beginning the negotiation of the Treaty to Versailles, which was supposed to end World War I, and he just faked it. He's like, I can't be sick now. So he just stuck it out and got that treaty signed. Of the U.S. soldiers who died in Europe, half of them were due to influenza, not to the war. There was a little jump rope rhyme that came out after this pandemic, and it goes, I had a little bird, its name was Enza. I opened up the window and in flew Enza. Sad, but a way to kind of, I guess, adjust. So here's what pandemics have occurred in our recent history. In 1890 to 1900, there was an H2N2. Remember, that's the surface proteins. And then the next one was H3N2, and then the really bad one came along, which was Spanish flu, H1N1. And then we had another pandemic in 1957, H2N2. And then we had H3N2 in 1968. That was the swine flu. I think this one was called, Spanish flu, Hong Kong flu, swine flu, I can't remember, Asian flu, swine flu, and this was swine flu. Sorry, don't try and remember those. I'm just goofing it up. So this was H1N1, and they were really concerned about this one when it came because it was another H1N1, and the target group was also the 20 to 30-year-olds. My daughter actually got that and got pneumonia, and went to the hospital, but she's fine. But yeah, another bad one. So here it is again, just in a little bit different terms. This pandemic, the Spanish flu, was in 1918. Then we have a huge gap of about 40 years, and we get in 1957, here it is, the Asian flu, with 1 to 2 million deaths. So this one was greater than 50. This one's 1 to 2. And this one's the Hong Kong flu in 1968, which is 700,000 deaths. And then we get up to the swine flu in 2009 with a little over 250,000 deaths. Now, you might notice there's a little bit of a pattern here. So this is 40 years. This is 10 years. This is another, 40 years, 35 years, something like that, 30 years. So these shifts occur, every 10 to 40 years. Oh, this one just got stuck in there. It's supposed to be at the end, so sorry. But recently the Spanish flu virus has been reconstructed from the tissue of a dead soldier and is now being genetically characterized. So they want to see get a little more information about the genetics of that organism, since it was so deadly, to hopefully shed some light on new and upcoming viruses, influenza viruses. So You probably know how it's transmitted. It replicates in the epithelial cells throughout your respiratory tract. It enters and exits through your respiratory tract, your mouth, your nose. Coughing, sneezing, and talking all spread it. The virus gains entry via inhalation and or direct contact or indirect contact, meaning, you touch a fomite, you touch a door handle, you open up the bathroom stall, and someone comes in right after you and touches the same bathroom handle, whatever. And it's recently shown that aerosolized virus can get in through the eyes. So, if someone coughs and, your mouth is closed and you plug your nose, it might still get in through your eyes. So it's just a possibility. And it can survive up to several hours at room temperature and in water, and it can survive months at low temperature, which is why it occurs in the winter months, the fall and winter months. It can survive in the air and on fomites and everything else much longer then. So what do we do to prevent it? You know this. You wear a mask, you wash your hands often, you cover your nose and mouth when coughing or sneezing, maintain social distancing, and you get a vaccination. So prevention. Health officials, how did they develop the seasonal influenza virus? So health officials like the CDC or the World Health Organization monitor what strains are circulating among people early in the year. And then they say, okay, we have an H3N1, we have an H2N5. And what they'll do is they'll choose some of those strains to make vaccine out of. So with a trivalent, that means 3. There are 2 types of influenza A. So in this example, H1N1 and H3N2. And then they add 1 type of influenza B as a kicker. There's also quadrivalent, meaning 4. There's 3 types of influenza A plus 1 strain of influenza B. So it kind of just depends on how many virus types are circulating as to what they do. So in case anybody asks, it is absolutely 100% not possible to get influenza from a flu vaccine. The vaccines contain either an inactivated virus, that means a killed virus, or they don't contain any virus at all. They just contain like the spike protein. So you can't get it. And that's a very common misconception. However, and here's where this part comes in, circulating strains don't always match the vaccine strain. Gosh. So let's say the strain has, we'll just take H, for example, it has an H3 and H4, and then it has a B. Well, the strain that's circulating might be an H6 or an H12, and so it doesn't match exactly. And that means you can still get it. So if you get it, or your friends get it right after getting the vaccine, they didn't get it from the vaccine, they got it from a different strain. So obviously the vaccine effectiveness is highest when the strains match. And even if it's a poor match, the vaccination will still cause you to cause antibodies, which confer some protection. Now, if you remember back to those antigenic drift and shift, there are different surface proteins. And if you get a vaccine, maybe one of the surface proteins is correct and maybe one isn't. So you do have some immunity. You may still get sick, but you do have some immunity still. And if you get the vaccine, most studies have shown that you will get less sick, and you are very unlikely or much less likely to get some of those dangerous complications that require hospitalizations and, getting pneumonia or having to be on a vent or something like that. So here's just a quick kind of cartoon of, the CDC throwing a hypodermic needle at what flu it might be. It's a little more scientific than that, but just a little humor. So how do we treat it? There's 3 antivirals. You've probably heard of Tamiflu. There's also Relenza and Repivap. These all work to interrupt the function of neuraminidase, which is one of the spike proteins, which when they interrupt the neuraminidase, they prevent the release of the virus particles from infected host cells. When they leave us, they grab some of our membrane and they put their spike proteins on. But if the neuraminidase isn't on there, then they're not gonna release it. They just can't ever get out. So here's also how we treat it. They say to consume water, clear liquids, take prescription antiviral medicines. It's important you get them within a couple of days if you can. If you can't, then you just take over-the-counter medications for your symptoms. It's kind of achy and headache and fever, etc. And then get plenty of rest. And very soon you will start feeling a little better. So I mentioned that I'm expecting, or I'm predicting another pandemic. I mean, all scientists are, if you know anything about viruses and influenza viruses in particular. When you get a new virus, an antigenic shift, you have a susceptible population. They've never seen it before. And what else does it require? So it requires a new virus. It needs to be transmitted from animals to humans, because remember how that shift occurs. It also needs to cause disease in humans, and then there must be efficient person-to-person transmission. So here we have a couple of them that have occurred. Antigenic shift occurred in all of these. And so now what we have in 2003, what appeared as an H5N1 avian virus. It's highly pathogenic in birds, especially chickens, and it has almost worldwide spread right now, Asia, Africa, and Europe. There's very few human cases, but for those who have close contact with chickens, there were about 230 cases and about 132 deaths. There is no efficient human-to-human transmission yet. So what do we need for a new pandemic? We need a new virus, which we have. We have a susceptible population. We have transmission from human to animals, and it can cause disease in humans, we can see here. And then it needs human-to-human transmission. So that has not occurred yet. So we do not yet have a pandemic, but we could pretty much any day. But it doesn't mean to panic. It just means you need to go get vaccinated. Tell your friends and family to get vaccinated. It's important because, as I said earlier, even if it doesn't completely match, it does provide some protection. So, all right, thanks. Bye.